ATP, liberating the second ATP molecule directly, at the same time forming pyruvate or pyruvic acid. Remember, at the stage of splitting, where we form the glycerate 3-phosphate, we have 2 molecules of glycerate 3-phosphate, meaning we end up forming 2 molecules of pyruvate or 2 molecules of pyruvic acid. Very interesting. Very, very interesting. In summary, glycolysis is a series of reactions in which a glucose molecule is broken down into 2 molecules of pyruvic acid. This occurs within the cytoplasm of a cell. It begins by glucose getting phosphorylated, a phosphate group added to it from ATP, giving off ADP and glucose 6-phosphate. Glucose 6-phosphate undergoes isomerization, forming fructose 6-phosphate, which is further phosphorylated, addition of a phosphate group from ATP, forming ADP, and fructose 1,6-bisphosphate, which is unstable. Therefore, it splits into 2 molecules of glycerate 3-phosphate, also called triose phosphate. Now, each glycerate 3-phosphate molecule is phosphorylated. Addition of an inorganic phosphate. It also undergoes dehydrogenation. Hydrogen atoms are removed. They combine with NAD, forming NADH2 or reduced NAD. Glycerate 3-phosphate, after undergoing phosphorylation and dehydrogenation, forms glycerate 1,3-bisphosphate, also called triose bisphosphate, that then undergoes dephosphorylation. A phosphate is removed, which combines with ADP, forming ATP and glycerate 3-phosphate, which undergoes another dephosphorylation, liberating a phosphate group that combines with ADP, giving off the second ATP molecule directly, and in turn giving us pyruvate. Therefore, what are the products of glycolysis? We have 2 pyruvate molecules. We have 4 ATP molecules directly, because 2 from one glycerate 3-phosphate and the other 2 from the other glycerate 3-phosphate. But we also have 2 NADH2 molecules liberated, one this side and another from the other molecule. Later we shall see that each NADH2 molecule liberates 3 ATP molecules from the electron transport system, giving us a total of 6 ATP molecules from NADH2 plus a total of 4 ATP molecules directly, hence giving a total of 10 ATP molecules given off in glycolysis. But when we subtract the 2 ATP molecules that were used, we remain with a net yield of 8 ATP molecules from glycolysis. Therefore, we have a net yield of 8 ATP molecules from glycolysis. So products of glycolysis: pyruvate, NADPH2, then ATP. So that is a summary of glycolysis. Now, what happens after glycolysis? Remember, there are 2 types of respiration: aerobic and anaerobic. Aerobic occurring in presence of oxygen, anaerobic in absence of oxygen. Now, in absence of oxygen, anaerobic respiration, a form of anaerobic respiration known as fermentation, now occurs. Meaning, fermentation occurs if there is no oxygen. Therefore, pyruvic acid pyruvic acid will form products, namely alcohol in plants and lactic acid in animals. That whole process is what we are calling fermentation. In absence of oxygen, the pyruvic acid or pyruvate is broken down into alcohol in plants and lactic acid in animals. That whole process is what we are calling fermentation. Therefore, fermentation is the incomplete breakdown of organic molecule, releasing alcohol in plant cells and lactic acid in animal cells. Why do we call it incomplete? Because some energy remains trapped. Therefore, the organic molecules are not broken down completely. Fermentation is of 3 different forms depending on where or which organisms it occurs. Fermentation can occur in plant cells, it can occur in animal cells, it can occur in some bacteria. Therefore, when it occurs in plant cells, it leads to formation of alcohol known as ethanol. Therefore, that form of anaerobic respiration, of fermentation in plants, is what we are calling alcoholic fermentation, because it leads to formation of alcohol. This usually occurs in plant cells and even in yeast cells. Remember, how does this occur? After glycolysis, in absence of oxygen, the pyruvate or pyruvic acid undergoes decarboxylation. Pyruvic acid, in absence of oxygen, in plant cells or in yeast cells, undergoes decarboxylation. A carbon atom is removed and given off as carbon dioxide gas in presence of an enzyme called pyruvate decarboxylase enzyme, leading to formation of ethanol. So pyruvate undergoes decarboxylation, removing carbon dioxide in presence of pyruvate decarboxylase enzyme, forming ethanol. Ethanol is then reduced to ethanol. Remember, when we talk of reduced, that means hydrogen atoms are added. Reduction could be the addition of hydrogen atoms. So where do the hydrogen atoms come from to reduce ethanol? NADH2, nicotinamide adenine dinucleotide reduced, donates hydrogen atoms that reduce ethanol to ethanol. That is why we say, in summary, alcoholic fermentation in yeast and plant cells involves the pyruvate or pyruvic acid, which was from glycolysis, undergoing decarboxylation, giving off carbon dioxide gas in presence of pyruvate decarboxylase enzyme, forming ethanol. Ethanol is then reduced to ethanol by addition of hydrogen atoms from reduced nicotinamide adenine dinucleotide, giving off NAD and ethanol in presence of alcohol dehydrogenase enzyme. However, ethanol is toxic, and when aerobic conditions return, ethanol is oxidized to acetyl coenzyme A, which is fed into the Krebs cycle. So we may get a question asking us that what is the fate of ethanol in plant cells or in yeast cells? What is the fate of ethanol in plant cells on return of aerobic conditions? We're noting it is oxidized to acetyl coenzyme A, which is fed into the Krebs cycle. We are going to see what happens to acetyl coenzyme A. The second form of fermentation is lactate fermentation. The other one was alcoholic fermentation because of the product alcohol in plants and yeast. This is lactate fermentation because of the product lactic acid occurring in animal cells. Therefore, in animal cells, they respire anaerobically. Giving off lactic acid. How does it occur? The pyruvate or the pyruvic acid from glycolysis is reduced. In other words, hydrogen atoms from NADH2 are taken up by pyruvate, forming lactic acid, which lactic acid should not also accumulate. Therefore, what happens? On return of aerobic conditions, lactic acid is transported from the muscles and taken to the liver. Muscles to the liver, where part of it is converted to, pyruvate. Lactic acid transported from muscles to the liver, where in the liver it is converted to pyruvate. Now, some pyruvate is oxidized to carbon dioxide and water, while the remainder is converted to glucose. Now the reverse of glycolysis, this time from pyruvate back to glucose. Some pyruvic acid is converted or oxidized to carbon dioxide and water, thereby getting rid of the lactic acid. Another pyruvic acid is converted to glucose and stored as glycogen, because animal cells store carbohydrates as glycogen in the liver and muscle. While another portion of pyruvate is fed into the Krebs cycle. We are going to see what happens to pyruvate in the Krebs cycle, that is, in presence of oxygen. In summary, lactic acid, after being formed, converted back to pyruvate, which pyruvate, some of it is oxidized to carbon dioxide and water, another portion converted back to glucose, another portion is fed into the Krebs cycle. Lastly is acetaldehyde fermentation. This occurs in anaerobic bacteria, where pyruvate is decarboxylated, carbon atom removed, forming acetaldehyde, giving off carbon dioxide. Remember, alcoholic fermentation, because the product is alcohol. Lactate fermentation, because the product is lactic acid. Acetaldehyde fermentation, because the product is acetaldehyde in some bacteria. So that is what happens to pyruvic acid under anaerobic conditions. Common question. Describe the fate of pyruvic acid in absence of oxygen, or describe what happens to pyruvate in absence of oxygen. We could get another question. Describe the process of anaerobic respiration in yeast, anaerobic respiration in plant cells, anaerobic respiration in animal cells. What does that question require? Much as we've said that respiration occurs in those three stages: glycolysis, Krebs cycle, and electron transport system. Krebs cycle can only proceed in presence of oxygen. Therefore, in absence of oxygen, after forming the pyruvate from glycolysis, then fermentation will take place. So if the question is anaerobic respiration in plants, after talking about the glycolysis, formation of pyruvate, then I'll talk about alcoholic fermentation.